Thank you so much, Manuela. That was really amazing research. Next up, we have Emma Stierhoff with a presentation titled Assessing the Long-Term Impacts of Chronic Infection with Avian Malaria in Hawai'i Amakihi.
Aloha maikako. My name is Emma Stierhoff, and today I'm going to be giving a synopsis of my master's thesis research entitled Amakihi Kaulani Kaho'olomai, looking at the long-term impacts of chronic avian malaria in our Hawai'i amakihi. So Hawaii is home to a number of endemic forest birds. Over time, about 55 different endemic honey creepers evolved here in Hawaii, and they closely co-evolved with many of our plants here, namely the lobelioid family. So you can see in these pictures how close those tubular flowers match the bill shape of each of these birds. And so these birds and plants are heavily dependent on one another. The plants need the birds to pollinate them, and likewise, the birds need the lobelioids for a food source. Unfortunately, over time, honey creeper populations have declined here in Hawaii. Of the 55 species that used to exist in Hawaii, we now only have about 17 honey creepers still existing in the wild. Threats that they are facing largely include habitat loss, predators like cats and rats, and also disease like avian pox, mange, and avian malaria. Perhaps the worst of these is avian malaria, Plasmodium relictum. This is a protozoan parasite that targets the red blood cells and the liver of its host, and it's carried by the southern house mosquito, or Chlorodrapanus virens, which was introduced by Europeans in the 1800s. The acute stage of avian malaria, which is when the parasite is most abundant in the host blood, and so that most intense stage of infection, can cause a number of side effects for the host. This can include weight loss, anorexia, where a bird stops eating as much, enlargement and discoloration of the liver and spleen, anemia, which is basically extreme fatigue when the body's not circulating enough oxygen, and in worst cases, mortality. So this all looks pretty bad, but enter the Hawai'i amakihi, which is a beacon of hope for us in the fight against avian malaria. This little honey creeper has proved to be more resilient against infection with avian malaria than many of our other native birds. And so there's a lot of work being done to figure out what it is that makes them more resilient. Their mortality rates range from 2.5% to 65%. That's a pretty broad range, but this is largely based on where their population is. Generally, lower elevation populations have lower mortality, higher elevation populations have higher mortality. For comparison, apapane have a mortality rate of about 63% to avian malaria, which is higher on average, and they're rarely found below 250 meters in elevation, whereas amakihi can be found as low as sea level. And then the worst hit of this group is the EEV. They have about a 90% mortality rate to avian malaria. So they're rarely found where there are mosquitoes, so they're typically at higher elevations, such as above 1,250 meters. So chronically infected amakihi are amakihi that survive acute infection and maintain low levels of parasite in their blood, potentially for the rest of their life. There can be benefits to this, namely that it allows the Hawai'i amakihi to develop anti-malaria antibodies in their blood. And so if they are bitten by another mosquito with Plasmodium relictum, they're likely to avoid that acute stage of infection again. However, there can also be costs to this chronic infection with avian malaria. Not much research has been done here in Hawaii, but research on other birds has found reduced energy stores, like reduced fat or triglyceride levels. Also reduced hematocrit and hemoglobin, both of which can lead to anemia, or basically extreme fatigue. Also increased rate of telomere loss, which can relate to an increased risk of age-related diseases. Reduced activity levels, and also reduced reproductive success. However, in terms of reproductive success, that is the one study that has been done here in Hawaii looking at chronic infection in Hawai'i amakihi, and it actually found that nesting success was higher in pairs that had chronically infected males. So that doesn't seem to hold true for our Hawai'i amakihi based on that study. So with the study, I had two main goals to look at the long-term costs of avian malaria in this population of Hawai'i amakihi. My first objective was to assess the physiological status of amakihi with chronic avian malaria. And with this, I hypothesized that chronically infected amakihi would suffer greater physiological costs than uninfected amakihi. And my second objective was to determine if the concentration of Plasmodium relictum in the amakihi blood would influence their physiology. And with this, I hypothesized that a higher concentration of Plasmodium relictum would lead to a reduced physiological health in the Hawai'i amakihi. So how do you assess physiological condition? There's a whole plethora of variables that you can look at that tell you physiological status of an individual. I decided to look at just four that I thought would be most informative for avian malaria. So I first looked at hematocrit, which is the proportion of whole blood that is red blood cells. So this is a good indicator of how much oxygen the body is able to transport. I also looked at oxidative damage. Basically, in an immune response, your body creates something called reactive oxygen species, and this targets foreign agents that could be harmful to the body, but in the process, it also attacks your own body. And so oxidative damage is the damage that results from those reactive oxygen species. I also looked at triglycerides, which are a good indicator of nutrient levels, and body mass. And so with all of this, I would expect a worse physiological condition to be associated with lower hematocrit, higher oxidative damage, lower triglycerides, and lower body mass. So my study site was at Kauehu, which is presently known as Ainaho Ranch in Hawaii Volcanoes National Park. It's in the moku of Ka'u in the Ahupua'a of Keaupo. It's at an elevation of about 1,050 meters. It's an ohia-dominant open mesic forest. And the rate of chronic avian malaria for our Hawai'i amakihi there is about 32%. Ka'u has a really deep and rich history that I could do an entire separate presentation about, but it has been nurtured by Kanaka Maoli long before the establishment of this national park. And I'd like to draw attention to this Olelo no Eau, Ka'u Makaha, or Ka'u of the fierce spiders. Ka'u is a really harsh landscape. There's not a ton of farmable lands. There's a lot of areas that are really dry with harsh conditions. But the people of Ka'u are known for their ability to not only thrive, but persevere despite these harsh conditions. So for this study, I did sampling between the spring of 2022 and fall of 2023. We set up mist nets and caught birds in the mist nets and carefully extracted them for processing. Back at the banding station, we banded new captures with the USGS aluminum band. We measured the tarsus, bill, and wing length of each bird, and then we determined the age and sex based on their plumage, size, and breeding characteristics. Next, we took a 75 microliter blood sample from each bird, and then we weighed them, gave them some Necton, which is basically sugar water. Think of it like a lollipop after your doctor's appointment, and then we set them free. And at the end of the day, we took all of the blood samples back to the lab. We centrifuged them down to separate the red blood cells from the plasma. From there, we could measure the whole blood and the proportion of the whole blood that is red blood cells. That gave us our measurement for hematocrit. We extracted the red blood cells and stored them with blood buffer, and separated out the plasma and stored that separately. Next, for the lab methods, we wanted to quantify or determine the infection status of each bird using qPCR. So we first had to extract the DNA from each blood sample using this DNeasy blood and tissue kit. We then used qPCR to determine infection status. So basically, if the CQ value was 0, meaning that the parasitic DNA never amplified in these heating and cooling cycles, then malarial status was considered undetermined. So either the bird does not have avian malaria, or it's so low that it can't be detected by qPCR. And then if the CQ was between 20 and 50, the bird was considered positive or infected. I also measured reactive oxygen metabolites, again, that result of reactive oxygen species attacking the body. And I did this using the DROMS test from Diacon International. And I also measured plasma triglycerides using the lab assay triglyceride kit. For statistics, I created a global linear model for each of my physiological parameters, looking at the physiological parameter as the response variable and disease status as the explanatory variable, also including some fixed effects in some cases like breeding status, age, and sex. And then I also conducted a correlation analysis to see if there was a relationship between each physiological parameter, so each of the 4, and the CQ value. And I did this only for infected birds, so all of those zeros for infection or for CQ value were not included in that analysis. So getting into some results, first looking at hematocrit, I didn't find a difference between positive and undetermined birds in terms of their hematocrit levels. So if there is any red blood cell damage from chronic infection, it seems that the birds are able to offset this, perhaps by increasing their red blood cell production. And then looking at the relationship between CQ value and hematocrit, there was no relationship between the two. So this suggests a good level of tolerance in Hawai'i amakihi to chronic infection, where regardless of the intensity of that chronic infection, their hematocrit does not change. Next, looking at oxidative stress, although it was slightly higher in positive birds than undetermined birds, this difference was not significant. And so this seems that even if the chronic infection is increasing pro-oxidant activity that could potentially cause oxidative damage in Hawai'i amakihi, they're able to offset that with maybe more circulating antioxidants. Looking at the relationship between CQ value and reactive oxygen metabolites, there was no relationship. So again, this suggests that they're pretty tolerant of chronic infection. Looking at triglycerides, again, there was no difference between positive and uninfected birds in terms of triglyceride levels, so it doesn't seem that chronic infection really impacts their nutrient levels, at least not in terms of triglycerides. And also looking at CQ value and triglycerides, there was no relationship between the two, again suggesting tolerance to that chronic infection. And lastly, looking at body condition, this is basically a measurement of mass that takes into account the body size of each bird. So my metric for body condition was the mass of each bird divided by the wing length to control for that body size. And so this graph is showing that body condition metric, and it was actually higher in chronically infected birds than uninfected birds. This is kind of unexpected. You would think that if they have this low level of parasite, they would be doing worse. My hypothesis for why this might be is that avian malaria kind of acts as a filter for these birds, where these birds that are unhealthy to begin with don't survive the acute stage of infection. And so these chronically infected birds are just leaving the ones that were able to survive because they were healthier off to begin with. And lastly, looking at the relationship between CQ value and body condition, there again was no relationship between the two. So this, along with the other physiological parameters, suggests that these birds are really tolerant of chronic infection. Regardless of the intensity of that chronic infection, their physiology is not really affected, at least not based on these parameters I looked at. So in conclusion, there are no apparent physiological costs of chronic infection in Hawai'i amakihi, which is a big win. I'm really happy to be able to deliver good news on behalf of the bird world. And based on mass, chronically infected birds might even be healthier. Perhaps this is because unhealthy birds die from infection, or maybe healthier birds are more likely to get infected for one reason or another. And these results also suggest that amakihi are very tolerant of chronic infection. So what does this all mean? There have been many studies on acute infection in Hawaii native birds, but this is one of the first to assess the long-term impacts of chronic infection. There was that one other Kilpatrick study in 2006 looking at reproductive success, but otherwise we haven't really looked at the impacts beyond the acute stage. So it's really important that we understand the full impact of this disease on our native birds. It's also important to understand what it is that makes amakihi so resilient. So understanding the full extent of that resilience will enable us to better understand that and potentially help other honeycreepers with that information. I'm also hoping this will guide some future research, maybe looking at other impacts of disease on honeycreeper fitness, and also looking at if physiology can predict survival of acute infection. That would help answer questions like why are chronically infected birds better body condition than uninfected birds. And lastly, this is another testament to the resilience of our Hawai'i amakihi. I think that Olelo no Eau Ka'u Makaha, or Ka'u of the Pierce spiders, applies just as much to our Hawai'i amakihi as it does to the people of Ka'u. So there are so many people to thank. My committee, my thesis advisor, Pat, who's here today. Thanks for listening to this again. And the many volunteers and vendors and folks who came out to the field site with us. A number of organizations that helped support this work. And also most importantly, the amakihi. They're the main contributors to this project, and it was such a joy to work with them, so mahalo to them. And if there's time for questions, I'm happy to answer questions, and also feel free to reach out to me over email. I'm always happy to talk about birds.
So are the chronically infected birds a reservoir for malaria? Or are they able to, produce enough? Is it the parasite load high enough that it can be transmitted from those birds to others?
That's definitely a concern, and it's kind of interesting because the non-native birds have kind of been seen as the reservoir, and they're the reason it's here in the first place, but they clear infection completely. And we're not really sure if amakihi do that or not. So one of the goals of this Idaho project, which is ongoing, is to see if there are instances of the Hawai'i amakihi getting rid of infection completely, or if they do live with it for the rest of their life. And then we can better understand their impact as potential disease reservoirs. Yeah, Nella.
Yeah, Emma, so some of those amakihi you caught more than once, right? Was there enough of a sample size of birds that were not infected the first catch and then infected on a later catch that you could actually compare changes in their physiological conditions?
Yeah, so I didn't do that for this study, but it's definitely something I want to look at. For this data set, I only included one sample per bird, so if we had recaps, I just used the bird with the best blood sample, since I needed a lot of plasma for these analyses. So that is a limiting factor, but yeah, definitely we have seen cases of birds that were uninfected and went to become chronically infected. And we've had some birds that have returned like 8+ times to this field site. So it seems like there's a resident population that we can track repeatedly. So that's definitely something that I want to look at with this data set. Yeah.
So this is a bit of a background question. You mentioned a study that said chronic males had higher nesting. Was there any information about chronic females?
Yeah, I think the females didn't have an impact. Like, regardless of infection status, it didn't really change if the female was chronically infected, which is interesting. But yeah, it was only the chronically infected males that had higher nesting success. Otherwise, there was no difference. And that was Kilpatrick 2006, I think, if you're interested in checking it out. All good? Okay, mahalo.